Hello, this is Matt Dean with A Plus College Ready, and today we're going to talk about proteins. Proteins are arguably the most important molecules in your body. Proteins are composed of the elements carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. Your body uses proteins to build and repair your tissues. You also use proteins to make enzymes. Enzymes help to catalyze most of the reactions in your body. Your body wouldn't work without proteins. Proteins also make up many hormones or cell signals in your body. They also make up many other important chemicals in your body. Protein is an important building block of your bones, your muscles, your cartilage, your skin, and even your blood. Proteins are large macromolecules. They're built from monomers known as amino acids. Amino acids are attached together via bonds called peptide bonds. These bonds are a type of covalent bond, which means they're pretty strong and hard to break. The reactions which connect these amino acids together are called dehydration synthesis reactions. You might also sometimes hear them called condensation reactions. When your body breaks down proteins, which it might during digestion, reactions called hydrolysis reactions will actually break apart the amino acids and separate them from each other. In biology, there are 20 different amino acids that are used to make up all of the proteins in life. Here's a diagram which shows the general structure of an amino acid. These parts of all 20 amino acids are exactly the same. This functional group, an NH2, is referred to as an amino group. This one over here, COOH, sometimes you'll hear it called an acid group. Oftentimes you'll also hear it called a carboxyl group. There's also a single hydrogen atom connected to the carbon. The part that makes the 20 amino acids different from each other is the functional group known as the R group. And we'll look at some example R groups on the next slide. In some cases, you'll see an amino acid depicted as non-ionized, not charged. In most cases, though, in biology, the amino acids take on a charge. The carboxyl group is called an acid group because very often this hydrogen atom breaks away and is released into the environment. When that happens, the carboxyl group looks like this. Also, the amino group can act as a base. It can accept a hydrogen ion, and when it does, it becomes NH3+. So these are both amino acids. This one depicts it in a non-ionized form. This one depicts it in an ionized form, which is in most cases going to be what it's actually going to look like in the body. You'll see in this diagram the yellow parts are the R groups of some different amino acids. R groups can be very simple. They might be a methyl group, a CH3, as we see on this slide. Or they can be very complicated carbon-hydrogen rings, like in phenylalanine. But what I want you to realize is that some of the R groups are not polar. They're nonpolar or hydrophobic. Other R groups are polar and hydrophilic. And this has a huge effect on the shape of the proteins that these amino acids are found in. I'll talk more about that here in just a second. Let's take a minute and talk about how amino acids are connected together in dehydration synthesis reactions. So first of all, let's draw in a general amino acid. So here's one amino acid. Let's draw another one over here beside it. You typically always draw the amino group, the NH2 on the left, and the carboxyl on the right. So when these molecules are being bound together, the carboxyl group will lose a hydrogen, and the amino group will also lose, and I said that incorrectly, the carboxyl group will lose an OH, and the amino group will lose a hydrogen. That frees up a binding site on both cases, on both molecules, and with the help of some enzymes, a bond will form between the carbon in the carboxyl group and the nitrogen in the amino group, this bond is a peptide bond. And since it involves the sharing of electrons between nonmetals, in this case nitrogen here and carbon here, it is a covalent bond. So a peptide bond is a type of covalent bond. A protein is made of many, many amino acids connected together via these peptide or covalent bonds. It's important to remember that proteins are made of multiple long chains of amino acids. These long chains of amino acids are called polypeptide chains. In most cases, several polypeptide chains wrap together to make a protein. The polarity of the amino acids in these chains helps to determine their shape. Polar amino acids are attracted to water, which is everywhere in a cell. So these polar amino acids usually end up on the outside edges of a protein. Whereas the amino acids with non-polar R groups, they're hydrophobic, they're water-fearing, they don't like water, and they usually end up causing a fold to happen that will push them toward the inside of a protein where they can be away from the water. So the polarity of the R groups of amino acids is one of the key factors in determining the actual shape of a protein. And a really important theme in biology is that structure in biology, in most cases, structure determines function. So the shape of a protein determines how it works. Structure determines function. And one of the key things that helps to determine that shape or that structure is the polarity of the amino acids or the R groups of the amino acids found in that protein. Alright, so some key points to remember. Proteins are made of amino acids. Proteins are macromolecules. They're typically made of several long chains of amino acids wrapped together. Each one of these long chains of amino acids is known as a polypeptide. Now, we talked a second ago that the structure of a protein often determines its function. We often talk about the structure of a protein on four different levels. One of those is primary, which means first, secondary, second, tertiary, third, and quaternary, fourth. So let's talk about each one of these levels of structure briefly. The primary structure of a protein refers to the linear sequence or order of amino acids in the polypeptide chain. Which amino acids are there, and what order are those amino acids in? This primary sequence is determined by the DNA. Really, DNA's only job is to code for the primary structure of your body's proteins. The primary structure is held together by covalent bonds. In proteins, these peptide bonds occur between the carboxyl group of one amino acid and the amino group of the adjacent amino acids. And they're formed during dehydration synthesis reactions. They can also be broken apart during hydrolysis reactions. Here we see the primary structure of a protein. Each one of the little ball-shaped objects represents an amino acid, and the little lines that connect them are the peptide bonds. Again, remember that the primary structure is determined by DNA. The secondary structure is the shape or conformation of certain stretches or strands of a polypeptide. So it's how is a certain piece of a particular polypeptide chain shaped? These are fairly local shapes, local conformations, and they're often dependent on hydrogen bonding. Amino acids are polar, so they can form hydrogen bonds. These hydrogen bonds are fairly weak, and we'll see later that they can be broken. There are two main types of secondary structures. They're not the only ones, but they're the two main types. They are the alpha helix and the beta sheet or the beta pleated sheet. And notice in this diagram, wherever you see two dots, that indicates a hydrogen bond. So these hydrogen bonds help to stabilize these secondary structures. And although each one of them is weak, there are lots of hydrogen bonds. So collectively, they're pretty strong. The secondary structure, again, is the shape of a particular segment of a polypeptide chain. It's often held in place by hydrogen bonding. tertiary structure is the overall three-dimensional shape of one entire polypeptide chain. This chain is usually large, it's bent and twisted into a particular shape that will give it a particular function. It's also in the shape that gives it maximum stability. The tertiary structure is stabilized by hydrogen bonds and also these bonds called disulfide bridges, which typically occur between amino acids called cysteines that are found in protein. The R groups of these cysteines contain the element sulfur. So if two neighboring cysteines get nearby, those sulfurs will bond and form a bond, a disulfide bridge, which will help to stabilize and hold in place the tertiary structure of a particular protein. So the orange, almost lightning bolt shaped objects in this picture represent the disulfide bridges. Again, a reminder, the tertiary structure of a protein is how is a single polypeptide chain shaped. And that tertiary structure is held in place by hydrogen bonds and also by disulfide bonds. The quaternary structure of a protein involves the wrapping together of multiple polypeptide chains. And how do those chains wrap and form the overall final specific shape of a protein? There's a variety of bonding interactions that help to hold the quaternary structure in place. These include hydrogen bonds, salt bridges, and disulfide bonds. Again, the quaternary structure is the overall structure of a protein formed by the wrapping together of multiple polypeptide chains. So here we see a protein formed by two polypeptide chains, one in dark blue wrapped together with the second one that's more of a lighter blue color. So let's also mention the process of denaturation. Denaturation is a process in which proteins lose either their quaternary structure, tertiary structure, and or their secondary structure. Because the shape of the protein changes, this means that its function is lost. Denaturation can be caused by several environmental variables. These can include the addition of heat, changes in pH, either more toward the acidic side or toward the basic side, changes in salinity, saltiness, and also the presence of certain heavy metal toxins like lead or mercury. Denaturation usually results when hydrogen bonds and disulfide bonds are broken, and this destabilizes the protein's shape. So denaturation is when the shape of a protein is changed. It can involve the quaternary, tertiary, or secondary structures, and because the shape is changed, the protein no longer functions like it's supposed to. Let's also talk for a second about protein synthesis. Remember that proteins are constructed from amino acid monomers, and these amino acid monomers are put together by an organelle called the ribosome. Ribosomes use directions from the DNA via the messenger RNA to put together the proteins. Some ribosomes are embedded on an organelle called the rough endoplasmic reticulum. Proteins made by these ribosomes usually are either transported out of the cell and used by other cells or other parts of the body, or they may end up in the cell membrane as like transport proteins or carrier proteins. Let's also mention something called the endomembrane system. It's a network of organelles which synthesize, package, and transport proteins through and eventually out of the cell. Typically, the endomembrane system is going to start with a ribosome, which synthesizes the proteins. The proteins are then dropped into the rough ER, or the rough endoplasmic reticulum. There, the proteins are usually bent into shape, they're modified, and they're often packaged into a secretory vesicle. Those vesicles will then break off the rough ER and typically move to an organelle called the Golgi apparatus. In the Golgi apparatus, the proteins will be stored and they'll be modified. Different functional groups may be added to them, and they're also stored. Eventually, the secretory vesicles will break off the Golgi apparatus and move to the cell membrane, where they will bump into that membrane. The phospholipid portion of the vesicle will become part of the cell membrane, whereas the protein that's inside that vesicle will usually be pushed out in a process called exocytosis or secretion. So here we can take a look at the endomembrane system, and we can see the rough ER. The little dots on the rough ER represent the ribosomes. So those ribosomes are making proteins. They're dropping down into the rough ER where they're shaped, modified, and packaged into vesicles. Those vesicles break off, and you can see each of these little vesicles eventually heading to the Golgi apparatus where they'll be stored, further modified, packaged. They will eventually break off again, bump into the cell membrane. The phospholipid covering of the vesicle becomes part of the membrane, helping it to grow, but the protein contents are pushed out, and that process is called either secretion or exocytosis. The endomembrane system is a conserved quantity in eukaryotes, so it helps to show that all eukaryotic cells are related to each other. Other proteins are synthesized by ribosomes that freely float in the cytoplasm of the cell. These proteins typically remain in the cell and help it carry out its day-to-day functions. These proteins are often folded into their functional states with the help of a structure known as a chaperonin. So to summarize some information about proteins. Proteins are arguably the most important chemicals in your body. They're components of bone, muscle, skin. They make up most of your body's enzymes. They are also involved in cell-to-cell communication. They help to make up structures like hormones. Proteins are large macromolecules. They're also charged. They're made of monomers called amino acids. We can see two of these amino acids being hooked together through either what's called a dehydration synthesis or condensation synthesis reaction. Chains of amino acids hooked together are called polypeptides. Most proteins are made of several of those polypeptides wrapped together. Remember that the structure of a protein determines its function. And we talk about the structure on 4 different levels. The primary structure is the order of the amino acids in the polypeptide. The secondary structure looks at how are certain segments of a particular polypeptide chain shaped. The tertiary structure looks at the overall shape of each polypeptide chain, and the quaternary structure looks at how do different polypeptide chains wrap together to make the overall structure of the protein. Denaturation is when the shape of the protein is changed, and that can involve changes in either secondary, tertiary, or quaternary structure. It can be caused by the addition of heat, a change in pH, a change in salinity, or even the presence of certain heavy metals. Hope that helps.